So I'm John Hooper from the Queensland Museum. I have with me Catherine Hall, who's a co-author on this paper, and it's about pumping sponges through the taxonomic pipeline. And it involves two projects, the Sponge Barcoding Project and the SpongeMaps Project. And all these little boxes over here are people who helped us get this thing flying. So I want to talk about 5 problems or troubles that we have with sponges and the solution that we've arrived at to try and resolve these problems, and then some brief conclusions. So number 1 problem is the sponges have an economic imperative. In other words, can't we just ignore them? And the answer is no, is because nearly 50% of all new approved drugs for cancer have been derived directly from natural sources. And of these, as you can see from these 3 graphs, in terms of cytotoxicity, antimicrobial, and central nervous system protective bioactivity, the peripheral stand out as being the dominant or co-dominant groups of all marine invertebrates and plants, marine ones that is. Here's an example of quinones, which is a cytotoxic compound. It's thought to be mostly produced by microbes, but in terms of sponges, the relationship between the sponge and the microbe that seems to be important. Over the past 20 years, the sponges have been shown to be almost, once again, half of all bioactivity of marine invertebrates has come from sponges, about 49%. You see the nearest one is the cnidarians, which is mostly the anthozoans, of which most of the soft corals and gorgonians are more bioactive. Within sponges themselves, there are various families that have a higher bioactivity than others, and there's some that aren't bioactive at all. And to summarize this, if you look at the number of known species of sponges compared to the number of known species of anthozoans, they're pretty close, but the number of unknowns are quite different. And the number of marine natural products, MNPs, is once again almost twice as much as any other marine invertebrate phylum. So effectively we can't ignore them. Within sponges themselves, there's not an even distribution between species diversity, alpha diversity, and bioactivity, which is the marine natural products. And similarly, if you look at the family level of sponges, the species diversity doesn't correlate with bioactivity either. So a greater biodiversity of sponges does not necessarily imply greater bioactivity, and the challenge presented by the unknown biodiversity remains significant. Once again, we can't ignore these beasts. Using a strange little state of knowledge index, I'm not quite sure how they arrived at this, but Costello et al. In the wind-up of the Census of Marine Biodiversity Knowledge, found that the 4 most bioactive groups of marine invertebrates, the cnidarians, the tunicates, the peripheral and the bryozoans, are also amongst the least known animals. Just a quick run through. Megadiversity is another problem. With silicious sponges, there's about 7,500 species known, described species that is. With the glass sponges, the hexactinellets, which are predominantly deep water ones, there's only 600 species known. With the calcareous sponges, there's about 700 species known worldwide. And with a new class of sponges just erected 2 years ago, the homosclerophorid sponges, about 100 species known. Over the Indo-west Pacific, over the past 30 years, all these little circles basically indicate, all the black dots are collection sites. All these little circles that clump them together are basically groups who've worked on marine natural products throughout the Indo-west Pacific. And you can see around the Fiji area, New Caledonia, and Solomon Islands, Vanuatu are actually quite strong targets for them as well. I won't go into the people here, but if this is being recorded, you can come and check the groups later. But there's only scratching below the surface. What you're seeing here is a collection from one sled from the seabed of the Great Barrier Reef. If you look at this map over here, every one of these dots is a pie chart. And when they're expanded, you can see that the composition of each of these pie charts is quite different between the reefs. So the blue ones are the sponges, and the green ones are marine plants, and so on. The conclusions was from the study that animals and plants are not evenly distributed on the seabed. From this particular study, 23% of all invertebrates processed were sponges, from which about 1,300 species or OTUs were discovered. Astoundingly, 2 of the top 5 most abundant sponges were new to science, and 4 of these top 5 most abundant sponges were not found on the adjacent coral reefs. And most of our knowledge of the Great Barrier Reef so far is based on the coral reefs, with little known of the adjacent shallow water seabed. So while our collections continue to rapidly expand, our capability to process them within our systematics lags significantly. And just to show you, the number of unknowns is the thing killing us, not the numbers of known species. So problem number 3 is a cosmopolitanism and morphospecies problem. Increasingly, the sponges that we think are the same are actually found to be cryptic sibling species. And in many cases, the genetic differences aren't manifested at the morphological level. In other words, they're ultra-conservative morphologically. And our current estimates of genetic diversity, species diversity are greatly underestimated. To give a good example, every one of these pictures here have been referred to Digitia herbaceae at one time in their lives. But when a large study was done across the Indo-Pacific on those things, which we thought were Digitia herbaceae, they found that in fact the polybrominated diphenyl ethers, which have an antiplasmodial activity, actually have a geographic distribution, and using an ITS gene, they correlated almost perfectly with those distributions of the chemicals. So not only confirms a taxonomic split between Digitia and a mythical genus Lamellodigitia, but also confirmed species boundaries of this thing called Digitia herbaceae and a likely coevolution of bacteria and sponge hosts. I'm going to skip this one just in interest of time. And the conclusion is, but what about the barcoding fragment? So trouble number 4 is the sponge barcoding and the CO1 barcoding fragment. This project, which I'll explain, basically looks at 3 aspects: the taxonomic barcoding, which is the one it was established for, but also applied barcoding and evolutionary barcoding based on CO1. We were very fortunate in 2008 to receive funding from the Sloan Foundation to participate in the marine barcoding of life, and we supplied about 20,000 specimens of sponges from our collections, extracted and exported those specimens to Munich, where they had a medium throughput sequencing facility called the Sponge Barcoding Project. And it's coordinated by the GeoBioCenter at Ludwig Maximilian University in Munich. It was launched in 2007. Its primary task was firstly assessment of suitable markers, and then establishment of a backbone sequence database. It was part of MARBOL, as I've mentioned, which was funded by Sloan. The museum supplied the extracts and the specimens of about 4,000 OTUs, or operational taxonomic units, morphospecies, another name for them, and the extracts were stored in Munich as well as in Brisbane. And that's the website there if people want to check it out. Straight away, the several immediate results was actually sponge barcoding. CO1 does work on sponges, but previously, because it's so morphologically conservative, people thought it was a waste of time, but in fact it's actually been found to be useful. And some of those papers, here's an example. One of the early results was it actually was able to differentiate two genera which were thought to be rubbish genera quite clearly, and also find geographical variants that correlated with the CO1 barcoding gene. It also, the studies done by the group in Munich in collaboration with our lab when they're both postdocs here actually found that they did provide a higher level indication of systematics of sponges, and it found that CO1 works quite well, at least at genus level. The barcoding data were very useful for phylogenetic exploration. And it also pointed out some problematic groups like the one I just mentioned before between those two, what were previously thought to be rubbish genera. When they did a mass analysis of those specimens that were extracted and screened, you can see there's a differential between the ability to amplify the DNA. You've got a couple down here at the bottom which have very low success rates, and a couple up here which have average maybe success rates. And compare this with the sequencing success, and I'll just show you. So you've got some low families down here that didn't have very good amplification but actually had pretty good sequence success, and vice versa for these groups here. But interestingly enough, these are also the most bioactive sponges as well. So in addition to the unknown of the CO1 in terms of information, there are things like DNases and contaminants, and it's still imperfect primers that we're dealing with. So barcoding, for sponges at least, is still learning to crawl. The applied sponge barcoding, this has been useful in looking at chemical profiles of sponges and just seeing whether in fact they're useful for chemotaxonomic exercises or not. In this particular case, there's no correlation between these terpenoid sterols, which are potentially ubiquitous in microbial communities, compared to, say, the halogenated bromine compounds, which are potentially sponge-produced and actually do have a taxonomic signal. And then there's, of course, there's the evolutionary barcoding using the CO1. It's actually showing that, in fact, there's endemic families originate from cosmopolitan families of sponges by showing that sponges colonize or invade areas at different times, and they're not, in fact, caused by isolation. I guess part of the biggest problem with CO1 is that in the ideal barcoding world, as Beth Mantle's been involved with these butterflies, you have a barcoding gap which makes it quite easy compared to the sponge barcoding world where there's an overlap, and this has now been published quite a lot. It's mostly due to the slow mitochondrial evolution in both, or in lower metazoans, and that includes cnidarians and sponges. And but despite those problems, basically the CO1 is still the best available mitochondrial marker to address population structural questions. Like the previous talk, we've been looking at what we call the Erpenbeck fragment. I'm sure someone else calls it something different, but it's towards the 3' end of the CO1. Like the previous speaker, these were amplified separately and sequenced separately, not in one run. But basically, the suggestion is to increase the CO1 for lower metazoans, including sponges, because the standard barcoding fragment doesn't display sufficient variability in many sponges, in other words, species resolution. So the Sponge Barcoding Project website has the usual data. In here we have the taxonomy, which is taken basically from worms and the Australian Biological Resources Study. The specimen data, which is the GIS information, comes from AusCAM, which is Amalgamation Museum Collections. The bibliographic data also stored on worms, and the Sponge Barcoding Project, actually the only thing it contributes really is the sequences there, which are in the public domain. And the two things missing, of course, is the morphology and images of where these sequences come from. So we're not just talking about AGCTs, but we're talking about biological entities. So this led us to resolving the thin taxonomic pipeline, where there's a significant lag between more effectively documenting the world's sponge fauna and the hundreds of thousands of unknown specimens already in collections, defining these within the Linnaean systematics and making unequivocal identities and distributions widely accessible. This gap is presently in the order of magnitudes. So here at the top we have the source of the pipeline, which is all the dots on maps or specimens, the GIS information, which is in OBIS and GBIF. The other end of the pipeline is actually publishing OTUs that lead to formal recognition of the taxa as species. And this is the biodiversity information gap, and it represents the challenge that we're trying to improve. So SpongeMaps was born, collaborations in the cloud. It works with Internet Explorer, but it works best on new browsers, such as Chrome and Firefox. It's based on a Caspio Bridge platform, which is essentially a Microsoft Access SQL engine. There's the URL there. Oh, sorry, it should be org, I've just been pointed out. That's not a, I'll go back. That should be www.spongemaps.org. Thank you, Kath, for pointing that out. There's 4 levels of access: public, member, editor, and administrator. The public access, there's no password needed. It's read-only, species released I'll explain what that means in a minute, to the ALA. Maps of Living Australia is about 670 species have public access, and it's mostly Linnaean taxa, in other words, described species, but because we're publishing chemical data and other information for which there's no species concept yet formalized, in other words, they haven't been described, we're also publishing OTUs, unknown species. And this is people have different opinions about this, but basically it's exposing potentially 5,000 OTUs or species concepts to the world community to encourage people to actually make them into species, because we're not going to be able to do it ourselves. The outputs, they produce what we call mud maps. They're basically guides to the identification, include images, morphological data, chemical smiles codes, and these are stored in SpongeMaps. I've got it right there, .org. Taxonomic data harvested from Australian Faunal Directorate and worms. Specimen GIS data are harvested from AusCan. Sequence data are harvested from the Sponge Barcoding database and GenMag, and they're all exported in a bundle to the Atlas of Living Australia on the first of every month as an XML file. And so within SpongeMaps you can do various things, such as you can search for things that are bushy, red and in Queensland, and it returns a number of species which are accessible to the public. These mud maps are exported to, as I said, the Atlas of Living Australia. As an example, you can click on to find that species we just looked at, Silicata singaporensis. It returns a mud map or a species page. It also returns all the images we've exported up there, the taxonomy, classification, records. It can tell you where they came from and what date they were collected. And then you can open these data up in the spatial portal on the ALA, which can map them in terms of dots on maps, but it can also overlay them with environmental layers such as marine ecoregions of the world. In other words, you can do some bioregionalization sort of work on it. You can overlay it with environmental data such as levels of silicates, salinity, sea surface temperatures, endemism, species richness, and so on. If you want to really get into SpongeMaps, you have to join it as a member, and I have the gatekeeper here in terms of Catherine, and basically in here you've got much more flexibility in terms of searches. There's also, as opposed to 670 mud maps released to the ALA, here you've got access to nearly 4,500 species out of about 5,000 species we're dealing with. So you can actually see all the undescribed species. Anyway, you can search for things like chemical compounds. It returns in, and you can even use this little widget here to turn the smiles code into a 3D chemical structure. You can search for DNA sequences, which basically are stored within SpongeMaps at the moment, but will be a live link eventually to the Sponge Barcoding database, and it'll take you to a particular thing on the database there. You can look at inventories from a particular area. Here we've got us looking for Heron Island, and it returns a number of specimens and species in Heron Island. You can download these data for yourself into a spreadsheet, a zip spreadsheet, and so on. You can add and edit new data. I'm just going to skip over these. You can look at various things like you can upload bulk data, such as this, from a spreadsheet to put information in. We can do it manually. There's all these other templates that you can do, and I encourage you, if you're interested, to go and have a play in there. And you can edit the data by adding morphological species descriptions and so on. And this links to worms. And finally is the use of statistics. This was launched in late April, and since then we've had about 860 visits, of which about half were new visits. So it's actually being used. You can see the map over here of the various geographical localities people of users have been coming from. So we're actually quite impressed with the uptake of this. And finally, the conclusions is we've been able to objectify the early process of sponge taxonomy by exposing the OTUs and including pinning sequence data and chemical data sets to those, so that even though there may be morphospecies, there are at least real biological entities that one day will become the main species, if, of course, there's resources to do that. They produce outputs for the sophisticated online biodiversity informatics portals like the Atlas of Living Australia and the Encyclopedia of Life. They give biological context to specimen GIS data points, or dots on maps, barcodes and chemical data sets, and they can move multiple collections of sponges and data sets into a collaborative online working space behind password protection that increases opportunities for collaborators to debate taxonomic identifications and so on. These are all the people who helped fund this. Thanks very much. And acronyms. And I'll just leave you with this interesting. It's going to play. So you thought sponges were sedentary, non-moving animals. This is a 6-day time-lapse high-definition video of a sponge being a sponge. Basically, we call it sneezing or coughing, and all those little things you can see coming off it are asexual buds. So while it's breathing and sneezing, it's actually reproducing. Thank you very much.
Thanks, John. Unfortunately, we couldn't see the video. Can you try to project it again?
No, it's I've got it embedded in the PowerPoint now, so it's obviously not going to work on this thing.
Switch to another screen on your, pull that drop-down menu and show another window on your computer and play it there.
Drop-down menu?
Yeah, the Show My Applications. Just pick another window in which you can show it. Or try showing it now.
Okay. Can you see that? Is that working?
No.
Okay. No, we'll have to abandon that.
Beth suggests you describe what goes on in the video.
Okay, so basically it's a 6-day time-lapse high-definition video of a spherical sponge called Tethia, and approximately every 4 to 6 hours, the sponge undergoes a massive contraction and expansion cycle, which looks like a sneeze or a cough. Its body size reduces or increases to about half, and at the same time it's actually moving on the seabed and producing these asexual propagules. Well, they're basically asexual larvae, and so the whole concept of a sponge being sedentary and just sitting there not doing anything is actually been turned on its head. So it's more for amusement than anything else.
Okay, thanks. If people have questions for John, please put them into the question window. John, if you want, you can open up a media player, a video player in another window and show it there. Okay. And then you just get out of PowerPoint, get into a media player. Any questions? And while that's going on, I ask our next speaker to get her PowerPoint ready to queue up and unmute her microphone.
Yes.
Okay, that's good. Hold on, Esperanza. Any other questions for John? John, I have a question for you, John. Do you have copyright access to SquarePants SpongeBob?
No, I don't. I'll have to remove those.
So you're infringing on copyright?
Possibly. That's from the internet.
Someone in the audience said one-time educational use might have the fair use exclusion. All right, I don't have any questions yet. You want to try to project the movie? While you're doing that, I do have a question. For the unnamed things, for the taxa that you haven't named yet, have you put the data in GenBank? That would be what Rod Page calls dark taxa.
Yeah,
so
okay, it's a good question. It's a good question. We haven't put the sequences of unnamed specimens, species, OTUs, whatever, in GenBank. We haven't yet put their morphological information on SpongeMaps. I'm showing the movie now on a separate screen. Can anyone see that?
It's not showing. Have you launched it? No.
Yes,
it's not showing.
Okay, I'll stop it. And it's a good philosophical question of we have 5,000 OTUs, of which only we think about 1,200 are actually within the Linnaean systematics. We know that some of these OTUs will never get described unless you describe them as sequence species, NTUs. And when we did that, would we actually want to give that information away? And I'm coming to the end of my career, and my answer is yes. But those young ones who are falling behind may have a different point of view. But it's a good question.